This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 876, recorded on March 15th, 2022. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Austin, Texas, Rich Condit.
Hello, Vincent. We have here, what we have is advertisements over my weather app. We have 74 degrees and sunny. It's windy, but it's nice, okay? And we're looking at nice weather for the foreseeable future. We may be out of the woods winter-wise, not that the woods are very thick here in Austin, Texas.
I was going to say, yeah. Not as thick as our woods. Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 46 Fahrenheit, which is almost 8 Celsius, and it's partly cloudy. We had some surprise snow the other morning, but it was melted by afternoon, and I keep thinking that's going to be the last snow, but we could have more.
Yeah, we had a little snow squall on Saturday, and it melted on Sunday. I was thinking the same. That's probably it. And from
I was in Boston on Saturday. Got the same squall.
Got the same squall, yeah. Interesting. Yep. And from Madison, New Jersey, Brienne Barker.
Hi, great to see everyone. I, in fact, was flying back to New Jersey during that squall, which was surprisingly unpleasant. But that snow is totally gone because it's currently 69 Fahrenheit, 21 Celsius here, and sunny and lovely.
Our guest today is from the University of Arizona, Michael Worobey, where I guess it's a little warmer. Welcome, Michael.
Thank you very much. Yeah, 79 Fahrenheit here. Might be saying goodbye to the 70s for 8 months here now, but great to be with all of you guys, and thanks so much for having me on.
No, my pleasure. The last time I saw you, you gave a talk at the Ian Lipkin thing, and
all right,
you said, on TWiV, you were talking about the HIV, the data tracking HIV origins through sequences, and you said, and listen to the TWiV on this. They did a good job of explaining it. I thought that was good. So here you are, and we have from you, you have your name on 2 preprints. Exploring the Huanan market as the epicenter of SARS-CoV-2. So let's talk about those. But first, let me ask you, what kind of training did you have to bring you where you are today? What kind of education and so forth?
Yeah, so I did my biology degree at Simon Fraser University in British Columbia. And I worked with a guy named Bernie Crespi there, who was a very, very good evolutionary biologist, really well-rounded, and he had an interest in phylogenetics. And so I became interested in phylogenetics at that point, and in disease, infectious disease and parasites, and then went to the University of Oxford and worked in a group that was sort of informally led by Eddie Holmes, and also included people that you might know of, like Andrew Rombo, Ollie Pybus, David Robertson, and so I kind of fell into this kind of golden era group of people combining the power of phylogenetics and the immense data and important sort of clinical side of things with viruses like HIV and dengue, and so trained in that sense at the intersection of the 2 disciplines. And then since then, I was very much a computational group. And then when I set up my own lab at the University of Arizona, I became interested in the sort of wet lab side of things to generate my own data. And so we've done work on pulling out gene sequences from hopeless old specimens that have been preserved for a long time, and you wouldn't think you'd be able to get HIV gene sequences out of them and that sort of thing to answer, when, where, and how did pandemics like HIV and Spanish flu start. And I've always tried to weave in where I could somewhat non-traditional lines of evidence, like what was happening historically in Central Africa at the time that HIV emerged, and do those sorts of extra angles actually enrich the more traditional scientific lines of evidence.
How long have you been in Arizona?
About 19 years.
Okay.
So when COVID-19 emerged, this was clearly something right up your alley, right? So you started working on it right away.
It was, yeah. I was pretty sick. I have chronic migraines, and I was in a real bad space right when the pandemic hit. And so for the first couple months, I was like curled up in a ball in a dark room and feeling like fantastic. Like I've spent my whole life training for this. And now I'm sitting here, just trying to get through the day. And completely incapacitated, basically. And then things cleared up a little bit, and I started sort of into this. And there's like a through line between the first project I worked on and these preprints that just came out. So the first thing I started working on was the emergence of SARS-CoV-2 into North America. This early outbreak in Washington State that may or may not have come from the WA1, the first patient that was diagnosed in the States, this Chinese national who had been to Wuhan and then flew back to SeaTac Airport on January 15th of 2020. And so, yeah, I had followed that story, and Trevor Bedford and the Seattle flu study people had done fantastic work generating genomes from late February from community cases. And it turned out that they were really close to the genome of WA1, just a couple mutations away, some of them. And that suggested to them and to me, when they laid out the evidence, that that epidemic probably had actually been cryptically spreading from this early patient who arrived on January 15th. Because, what are the chances? The chances were really low that this big outbreak would be so similar to that imported case, because there's a whole bunch of other branches on the phylogeny that it could have been. But the more I started analyzing the gene sequences and getting a feel for how the virus evolved, how slowly it evolved, it started to occur to me that maybe this clean break of two mutations between the virus that supposedly started it all and everything else that you see later might be a bit weird. And so I thought to myself, what we really need to do here is not just sort of stare at the patterns and say, that seems a bit weird to me. I don't think it was WA1, or it doesn't seem that weird to me. I think it was WA1, a single introduction. That just doesn't cut it, right? So what seemed clear to me is you need to model the outbreak. You need to run the tape of the outbreak in Washington State over and over and over again under the constraint that, we're going to start it on January 15th, excuse me, with the genome of the WA1 patient. So we're going to enforce that it happened with a single introduction. And we're going to simulate that outbreak over and over and over again. And we're going to evolve genomes into, February and March under that constraint. And then we're going to look at the patterns that we see in the simulated data and ask, are those, in the observed data, these patterns that we see, and not just the two mutation difference, but other patterns like, do you ever see another branch that's two mutations, but a different two mutations away, and then a big clade emanating from that, as well as the other two mutation clade? To make a long story short, the observed data was extremely unlikely under those simulations. And so it looks like actually it was a separate introduction, a weird coincidence of a pretty similar strain, but a second introduction. And so it was kind of cool because that WA1 patient could have gone down in history as another patient zero that, a lot of people were looking to blame Asian people of Asian ancestry for the whole outbreak, or I'm sure the North American outbreak. But we were able to show, no, actually probably, well, it wasn't this guy. And in fact, the timing of the second introduction suggests it was probably after the ban on travel, the ban on people from China traveling into the US. And so a good chance that actually that outbreak in Washington State was brought in by a US citizen or a visa holder who was allowed into the country with this incredibly lax oversight at the border, where you could just basically be asked, do you have a fever? Say no, and be waved through.
Wow. So we actually covered earlier on TWiV Without You, a version of the early story in the marketplace, and that's when I had emailed you, and you said, "Oh, we have some more coming out." And so that's what we have here. We have two preprints. The Huanan market was the epicenter of SARS-CoV-2 emergence, and you're the first author on a list of authors, many of which who have been on TWiV actually, including Christian Andersen and Bob Gary and Marion Koopmans. So
hang on, but while you're on the list of authors, this is a question for me. This is a remarkable list of authors from all over the place. How does that come together?
Yeah, interesting. So I had, there's two preprints here, and each has its own story, although there's a lot of overlapping authors. So for the companion paper that's more on the phylogenetics and the take-home messages, there were very likely two cross-species transmission events, one for lineage A and one for lineage B. That was a continuation from that paper on the emergence of the virus in Washington State, where Joel Wertheim and his graduate student, Jonathan Picard, developed some really beautiful, the beautiful simulation technology, and Nima Moshiri as well. And so that group sort of gathered a few more co-authors for that paper. Then the Huanan market epicenter paper, there was a sort of convergence of two different projects. So I put out a perspective, or Science put out a perspective that I wrote back, I think, in October of 2021, where I had been looking at the question about whether we should trust this impression that a good chunk of the earliest cases really were linked to the Huanan market. Or whether it was possible that the Huanan market, the impression that the Huanan market was a sort of dominating risk factor early on, was because there was a lot of focus on it, and cases elsewhere were being not ascertained. So, in scientific lingo, was this due to ascertainment bias? And so I started trying to figure out everything I could about the early cases in Wuhan in that project. What I found was, if you cut through all of the sort of downstream analyses that could have been biased and go right back to the firsthand accounts by the doctors who actually figured out that there was an epidemic and that it was linked to the Huanan market, because it was the doctors, not epidemiologists, not China's pneumonia of unknown etiology program, not the China CDC. It was doctors in Xinhua Hospital, Zhang Xishan in particular, who on December 27th looked at some CT scans from a couple who both had these terrible lesions, these ground glass opacities, that she had seen some similar stuff with viral pneumonia before. She had seen SARS-1 patients, but she'd never seen anything like this. And so on the 27th of December of 2019, she was pretty convinced there was a new virus in town. These two people were in their 60s, and their son was with them. He was fine, but she forced him to have a CT scan as well, because she thought maybe this was infectious disease. Turns out he was loaded with the lesions as well. So on the 27th and these people were not connected to the Huanan market, importantly. So on the 27th of December, she had figured out almost certainly this was a viral pneumonia, a new pathogen, that it was human to human transmitted, and that you could be asymptomatic, but still infected. It wasn't until virtually a month later that China actually finally said, "Hey, yeah, this is human to human transmitted," and actually shut down the borders of Wuhan. But over the next 2 days, 4 more patients she saw, and her team there saw 4 more patients. 1, 2, 3, 4 of them, all workers at the Huanan market. So they had an emergency meeting on the 29th of December, said, this is clearly an important epidemic. There's clearly a connection to the Huanan market. Now we're going to tell the Wuhan CDC and the Hubei CDC about this connection to the Huanan market. Epidemiologists didn't know anything about Huanan until that point. So this is a pure, unbiased look. The first 7 patients there, 4 of them were linked to the market. The other 3 were part of a family cluster. So it's, even more than a 4-to-3 proportion, really. At another hospital called Wuhan Central Hospital, although they didn't know, they hadn't picked up on the Huanan link themselves, retrospectively, over the same period from December 18th to December 28th, they also identified 7 cases. 4 of them retrospectively turned out to be linked to the Huanan market. So very clearly, a good half of the very earliest patients were linked to the market. So this is a real phenomenon. The early cases really were linked to the market. And that work then led me into trying to extract from this WHO study report that had been done in conjunction with Chinese scientists and public health officials, that was, let's say, suboptimal in its presentation. The very, very low resolution maps summarizing the 164 cases within Wuhan city limits, with dots 2 kilometers in diameter, that the density of cases mapped by home address was so high that you couldn't really make out where the cases were. There are too many sitting on top of each other to make them out. Nevertheless, in that paper, I started working on, can we get more information on the mapping, because this can sort of tell us more about this idea of early on, are the early cases linked to the market geographically? Because this is a key question, because fundamentally, and this seems to get lost in the Twitter conversations, fundamentally, you really need to ask yourself, if this didn't come from an infected animal in this market, which is one of 4 markets in Wuhan that sold the same sorts of animals like raccoon dogs that were involved in the emergence of SARS-1. Okay, there's only 4 places in a city the size of, bigger than New York City, 11, 12 million people. There's 10,000 other places, nursing home, where an initial cluster of a respiratory disease could be noticed. Nursing homes, meat packing factories, bars, stadiums, biogen conferences, on and on, right? It's very weird that the place that it shows up first is one of these places. And so anyways, long answer, I was working on the geography of it and found a way to actually extract reasonable information on the location of 156 out of the 164 cases in Wuhan. And I was off to the races and was able to start doing some actual analyses, and then found out that Christian had been leading an effort to do something very similar within the market and looking at the geospatial pattern of where environmental swabs that had been taken from surfaces after the market was closed on January 1st, about 1:00 in the morning, January 1st, 2020, samples were of things like the ground, open drain grates on the floor, where cages of animals sat and they shat into. In some cases, an actual cage, a feather remover. In some cases, surfaces that are very animal associated. In other cases, surfaces that are most likely human associated. The walls of a vegetable stall where there were human cases, but no animals being sold. And so it turned out that we had been sort of working at different scales on this similar question, and decided that the combined analysis would be much more powerful than either of these things on its own, and then started working together as a team, and started how these things work, where once you have a team of people together, you can do things that you just can't do without the group of minds together. And it's been like a very extraordinary privilege to be working with these people, and finding out some of this stuff with them. So yeah.
So that commentary you just mentioned, that's what we did on TWiV. So that's what started this whole thing whose story you just told. So now we have this first preprint. It involves a lot of spatiotemporal modeling. Maybe you could tell us what exactly you did that makes it likely that this was the epicenter. And by the way, you said it was weird that it was this market, but not really, because we know for SARS-1 the market was the epicenter, right? So it's not unexpected.
I only mean it's weird if you think that it didn't, if you think it came from some other source than the market. If you think, and certainly, I mean, we're all well aware that a lot of people think that this came from a lab accident in Wuhan, and in particular, people have pointed to the Wuhan Institute of Virology. And I have been at an earlier point, somewhat, I don't want to say taken in because it sounds pejorative. I have been lab leak curious. Not to the point where I ever thought it was more likely than a natural origin, but I did think, it does seem a bit of a weird coincidence that this outbreak starts in a place that has a lab that studies these bat viruses. I actually, this sort of famous science letter that several of us wrote that kind of appears to have motivated by the Biden administration to start its 90-day intelligence review. That was my, or at least I suggested the idea to the lead author, Jesse Bloom, that we maybe send something to science just to say, let's not prematurely just rule that out. Let's keep an open mind. And so I have had an open mind to this. And if the evidence had gone differently, I would be sitting here talking about how I think this came from a lab accident. But over and over again, the evidence is pointed the other way. So for example, at an earlier point, a lot of the focus with people who thought it came from a lab or the WIV was this Mojang mine, where this sample that used to be the closest to SARS-CoV-2, RATG13, was collected. And there were 8 other genomes from bat samples that apparently existed but hadn't been published by Shizhong Li's group. And I actually thought that was a bit weird. Why aren't they published? And is one of those samples, is one of those genomes very similar to SARS-CoV-2, and that's why they're not published. And I actually got in touch with Nature magazine and said those genomes sequences have been mentioned in an addendum to a paper that you've published, and from where I'm standing, those seem to be things that would fall under Nature's policy of you have to put your gene sequences in a public database. And so would you please get in touch with the authors and have them deposit them in a public database? Because I was curious about testing the lab leak theory. And within a couple of days, the editor had followed through on that very, very quickly and effectively. And those data were posted in a public repository in China. And I had a day or so looking at them before Shizhong Li actually put out a preprint showing that they're very boring. They're none of them are close to SARS-CoV-2. And there's kind of the evidence, again, just points away from that. And so I've moved on with the evidence. And now I've talked so long on that tangent that I'm pretty sure I've forgotten to answer your question.
So it's all fascinating. It doesn't matter. You should summarize the first preprint on the spatiotemporal modeling that you did to ascertain that it was the epicenter. Yeah.
Okay. So let's take the pattern of cases in Wuhan in December. Okay, so we have a before and after kind of picture here. From the WHO report, we have 164 cases where people had disease onset in December. They may have been hospitalized in January, but when they're hospitalized, they're asked, when did you first feel sick? And that date is in December. So this is, and we have the home address location, which I was able to extract from the maps and put into numerical lats and longs that we could then put into actual spatial analyses. And so that's the before. That's the early snapshot of where cases are in Wuhan. And then I was lucky enough to have access to an after data set. So where cases or likely cases were a little later in the outbreak in January and February. And these were users of an app on Weibo, a Chinese social media platform, where people could get in touch for help with COVID if they had COVID. So this data set was about 800 cases or users of the app who presumably had COVID, and it's with cell phone geolocation data. So I had cell phone geolocation data with where that person was when they asked for help all over Wuhan. And let's talk about the after first. In January and February, it's clear that this virus is all over Wuhan. And in fact, you can basically overlay the density of users of this app who have COVID on the map of the density of people living in Wuhan. It's a complete reflection of the high density in central Wuhan. And in fact, it's an even better reflection of the density of people who are over 60. Because who gets most symptomatic COVID? People who are over 60. And so that's very strong indication of, this is a virus that's everywhere. So then what does the before look like? Does it look like that? No, the cases are just strongly clustered around this little market that sells foxes, porcupines, hedgehogs, rabbits, and raccoon dogs, all things except maybe hedgehogs that have been shown to be susceptible and in some cases able to transmit SARS-CoV-2. And in it's in the paper, but I don't think I did a good enough job of really highlighting how important this is. The cases at the market that had been known were all of lineage B. Okay, so for your listeners who don't know all the details, early on there were two lineages of this virus that were identified. And just like the Washington State versus WA1 case, they differ by two mutations, lineage A and lineage B. Lineage A is more likely more similar to the ultimate bat ancestor. Lineage B looks like those two mutations are about a month of evolution sort of downstream of lineage A. So on the face of it, the fact that only lineage B had been seen at the Huanan market was a legitimate sort of question mark about whether it came from animals at the market. It looks like it's a derived lineage. And if lineage A was the real beginning of this in humans, why isn't lineage A at the market? Why is it just lineage B? So geographically, what we're able to do is for a couple of cases for both lineage A and for lineage B, cases that were unlinked to the Huanan market, which are particularly of interest because the ones that are linked to the market, we already know they're linked to the market, and they're less interesting and informative. Cases where they've been investigated, they've said, no, I don't work there, I don't shop there, I've never been there, don't know anyone who's been there. Those are quite interesting because if as they're also closely linked to the market geographically, it suggests that that's where the community spread first started. So back to lineage A and lineage B. What we found was for the two lineage B cases where we had, where we knew the genome was lineage B and we had geographic locations, they were significantly close to Huanan market. Not too surprisingly, because lineage B we knew had an association with the market. They were about, I don't remember exactly how far away their median distance was, but I remember for lineage A. For the lineage A, in a city where you have cases that are sometimes 30 kilometers away from the market, the two lineage A viruses were only 1.17 kilometers away from the market. Now, if lineage A is not geographically associated with the market, if it didn't get its start at the Huanan market, these two early lineage A genomes have no business being found 1.17 kilometers away from that market. No business at all. That just makes no sense unless lineage A also has a geographical association, got its start at the market and started seeping into the local community from there. So that's a hugely important piece of this study that locks in with the other study where we find strong evidence there's only about a 3.5% chance or 3.6% chance that this was a single origin. So in other words, overwhelming probability that there was a separate origin for lineage A and lineage B. And if that's the case, what emerges here is a picture of lineage A and lineage B both emerging at the market, because we've got now this genomic geographical information telling us that lineage B also, not just lineage A and not just lineage B is geographically linked to the market. And it answers one more mystery about this outbreak. If lineage A was the actual single origin ancestor of this outbreak, wherever it came from, whether it was at the Huanan market or not, and it's roughly a month older than lineage B, which is 2 mutations downstream, then the question is, why is lineage A the minor variant? Lineage B is found in twice as many early cases in China as lineage A, which is odd.
I have a question about that. Going forward, did B then predominate, or was it more equal over time?
B became almost the only game in town. And so, when D614G swept through the country and Europe in the spring of 2020, that was the end of almost all lineage A. And that's because D614G had a pretty strong competitive advantage.
And that was in a lineage B background. Is that the idea?
Yeah. That particular D614G spike mutation as Betty Korber showed and was shown to be right, just made it a much more fit variant. But early on, before the D614G, there's not a lot of indication that the other lineage B was more fit. But in the Two Jumps paper, what we found was it does look like lineage B actually jumped in to humans before lineage A. And that's kind of like makes your head kind of not feel right, because lineage A is the more ancestral virus. But if these viruses jumped from a reservoir where there was at least a little bit of genetic diversity, there's no reason why a more derived virus like lineage B couldn't jump first. We know that these viruses, ancestral haplotypes, can be maintained for months. Okay, so in Washington State, for example, the sort of original variant of the second jump, that genome, you could find that same genome 2 months later in someone. Meanwhile, you could find variants that had evolved 6 other mutations. So anyways, the evidence points to, and these are not proven, but there's strong evidence that lineage B jumped before lineage A. And all of that led us, the whole picture led us to conclude, okay, we think lineage A was actually at the market. We think it jumped later than lineage B, and therefore we think it will be found, but at lower frequency than lineage B. And so as we were finishing dotting our I's and crossing our T's, this preprint from George Gao at the China CDC comes out. And although you don't see anything in the title or the abstract, and I think Christian Anderson and I were the first people to download it, I'm scanning it, reading it, and there's this line just buried in the middle of it, saying essentially, oh yeah, by the way, we found one lineage A. And I was like, literally, this is what I was like.
Smoking
Holy
gun.
Fuck, they found lineage A.
Where did they find it? Where was this?
In the market, right?
In a stall
okay.
Very near where in a seafood stall, but close in the same corner of the market where the most of the live mammals were sold. Now, this sample was taken from a glove. It was collected on January 12th, not on January 1st. But to me, and some people have pointed to, it's January 12th, so maybe it's not actually a real, from the market sample. It was something that was brought in later. But, the market was shut down on January 1st. It's basically a time capsule from that point on. It's not like people were coming in and going to work on January 11th and dropping their glove. The glove is, presumably there sitting from January 1st onwards. So it's effectively telling you what was there at the time the market was closed. So it really, it's clear that lineage A was also at the market as we predicted. And that was one of those pretty amazing moments that you don't get too often.
So based on what you're saying, I come away with the picture that both of these viruses were present in animals in the market for at least a month before the outbreak. First of all, is that a reasonable conclusion? And second, how would that happen? Is there one animal that spends a month in the market and has both viruses, or is there a transmission chain?
Sure. So here's a good point for me to say, first of all, which I probably should have said at the beginning. These papers are under peer review as we speak. And so it will be interesting to see what the peer reviewers see. And I'm sure there will be revisions. And so that's one thing. The other thing to say to what you just asked is there are lots of questions still unanswered. We don't know what animal was involved. I would agree that the fact that the Huanan market was the epicenter, it's almost absurd to posit that the virus came from something other than an animal or animals that were sold there. But we don't know that. We have, I should mention that we did statistical analyses that are based on some assumptions that for example, that when they did the swabbing of stalls, that they took the same number of swabs in each stall. Under that assumption, we've shown that actually having proximity to a stall that sold wild mammals was a more significant predictor of where an environmental swab positive sample would be located than where a human case was taken. And so there, and so that's important. It's also important that, people who have been proponents of the lab leak theory have been saying for a couple of years, not just that wild live mammals weren't sold at the market, but that these animals weren't sold at the market and that sales don't happen in the wintertime. We actually provide photographic evidence from December 3rd of what was there. So yes, I think what this shows is sustained sales of wildlife over a period of time, such that you get these multiple jumps that are spaced out in time. And you really are challenged to explain the combined phylogenetic evidence and the epidemiological evidence, the prevalence of lineage A versus lineage B, without this sustained presence of a source over time, which really seems only explicable by animals in the market.
And multiple animals.
Multiple, not necessarily multiple species, but multiple.
Yeah. Like I said,
you know what? I don't know if you've seen the pictures in our paper, but there are raccoon dogs stacked on muntjac deer, marmots stacked on deer, porcupines in the same room as red foxes. I would imagine there was plenty of animal to animal. I would imagine multiple animal species were infected, and that these viruses were present across multiple species for multiple weeks at least. And so, one scenario is you get the stars aligning and you have a bat virus with a human ACE2 using spike that rains down on a raccoon dog farm, which there were lots of in Enshi prefecture in the western part of Hubei province, hundreds of thousands of animals. It maybe is one mutation away from having a functional furin cleavage site. That mutation arises in one of the infected animals on that farm. It's now a very, very good respiratory mammalian pathogen. But it's a needle in a haystack. It's one farm. But animals are moved from that farm to Huanan market over a course of weeks as this outbreak goes on. And then once you have that, the experience of, Marion Koopmans has shown on farms where mink are infected, you have multiple cross-species jump from minks to humans on the same farm. 5, 6 individuals on the same farm are going to be infected or have been infected by those mink. So the market would be no different. Once you have, multiple animals over a period of time at the market, some people have said, it would be weird to have no jumps of this virus and then all of a sudden 2 jumps at the same place. Well, no, actually, all of the unlikely stuff happens before the animals get to the market. Once they get to the market, it would be weird to not have multiple jumps because these are highly contagious.
Is it then possible that there were multiple jumps, but that only 2 of them really succeeded?
Absolutely. And we actually evaluate that in the 2 jumps paper, that we show that the median number of jumps expected to give rise to the 2 successful jumps that we infer is 5. And the range, the 95% confidence interval does go down to 2. So it is possible that there were just 2, but it also goes up to 15. And the epidemiological data are also consistent with several more than 2 successful jumps. So there could be other successful jumps, but they're identical to lineage A or lineage B, and so they don't appear as
you can't distinguish them.
Yeah. And one other thing I should mention that is important here, that the gene sequences themselves have an important story to tell aside from all of the epidemiology. We find that with lineage A and lineage B, the time to the most recent common ancestor that you estimate is just with the gene sequences is around December 10th. So there are lots of different angles here that show that this virus was not around a long time before. We date the first primary case, which was likely a lineage B case, to late November. This virus doesn't seem to be plausibly present before November.
In people, right?
Beg your pardon?
In people, but it's obviously in some animal in the countryside.
In people, yeah. And it's in animals sometime before that. Although I would guess not too long before, because if there are 2 separate jumps, they are being drawn from a pretty shallow genetic diversity, like you might find on a single farm outbreak. But yeah, we need a lot more work on tracing the supply chains to these markets, and hopefully sampling was done. But you're not going to be able to test a few raccoon dogs that were collected around the time of this with, test them for PCR and expect to find much. I think you would really need to do serology, would be the most powerful way of doing this. And it may be that if this was a transient event on one farm, and what we do know is that the farms were liquidated early in 2020. Very early on, all of the civet and raccoon dog farms seem to have been, hundreds of thousands of these animals were culled and eliminated. We may have lost our chance to find these animals. And this is very different than SARS-1. With SARS-1, the market stayed open, the farm stayed open. We had years to find viruses in the intermediate hosts. In this case, a lot of has been said about 80,000 animals have been tested, and none of them were found to have SARS-CoV-2. Well, most of those were irrelevant animals like pigs and chickens. Not a single raccoon dog, for example, from the wild in Hubei or from a farm in Hubei has to date been reported on as being tested for this virus. And I don't want to get too focused on raccoon dogs, because as we show in the paper, there are a whole bunch of other species that were there as well, like foxes that are just as plausible. It may be that what we've got now is as close as we're going to come in some ways. And it's not, it's not bad. The John Snow and the Broad Street water pump, it's not bad. We use it as kind of the symbol of some great science. And in many ways, the mapping here, I don't want to try to sound like we're Jon Snow, but in terms of the evidence, I do hope that people who are used to or who expect gene sequences to be the be all and the end all here will open their mind to the fact that there are other kinds of data and knowledge that can be very, very powerful. And we have them in this case. And I would argue we have them in this case, like the geographical and the epidemiological evidence. For example, the stuff I was saying about there's no ascertainment bias issue. A huge proportion of the earliest cases really were people who worked at that market, and the rest are probably one or two transmissions away because of asymptomatic cases. You don't have that for many other emerging viruses. And so what we already do have for this virus, although it's not as good as in some ways a virus in an animal that's directly connected to a person, in other ways it's better evidence.
So go ahead, Marianne.
So Kathy had asked a question a bit ago about the frequency of emergence, that it may have been, 2 or more than 2, and you mentioned the data that you have on that. Is there any way to connect that with the urban location versus perhaps a more rural location of the farms? Does the location and the population density matter at all?
Yeah, that's a great question. So let's say it did originate on a farm, and the possibilities, most likely possibilities, seem to be a farm, probably in Western Hubei province, or wild caught animals, also perhaps even just surrounding Wuhan or locally in Hubei. If it was a farm, let's say in a rural part of Hubei province, I don't know what the PPE practices would be on a farm like that, but I would guess that if there had been an outbreak on a farm there, that there probably were cases of human animal to human transmission on that farm, but that they likely didn't start a successful chain of transmission, because as we showed in PCAR 2021 in Science, 99% of those cases don't start a successful chain of transmission. And it wasn't until, if it did start on a farm, it wasn't until those animals got into a tinderbox of one of the largest cities in China that it took off. But yeah, I think there were almost certainly several cross-species transmissions of this virus, including ones that happened outside of Wuhan.
So would it be worthwhile to take, I mean, the raccoons' dogs may be gone, but the farmers are presumably still there. Should we get some serum and check for antibodies from them? Would that be useful? Is that too noisy?
Too late to collect serum now, because it will be confounded with subsequent, they could have been infected with SARS-CoV-2 between now and then. Archival samples are always, it's something I've done work with HIV, and I think those are always interesting to think about. The question is, there has been a pretty concerted push on the part of Chinese authorities on this narrative that the pandemic started from somewhere outside of China. Even in Gao et al.'s recent paper, the final paragraph talks about serological studies suggesting that the virus might have been in the US or Italy as early as September of 2019. Now, these are not credible studies in my view, or the view of, I think, anyone who understands the early epidemic. But the final paragraph talks about those and how we should have an international effort to look outside of China. And so there may be difficulties in actually getting at some of these questions, but the fact that paper just got put out as a preprint, stuff's still coming out, and we have been working behind the scenes, particularly Eddie Holmes, who knows some of the co-authors on that paper. We have been aware of that work, in part because we got this leaked version of that of much of that that we used for our study, that was then confirmed by Gao et al.'s preprint. So we've been aware of that preprint and the importance of the metagenomic data from some of those samples. And by that, I mean, for your listeners who might not be familiar, with when you swab a cage and then you generate a viral sequence, the viral sequence is one thing. But if you generate that sequence by using a technique that we call metagenomic sequencing, you're basically sequencing all of the RNA that was in that sample. And that then allows you to see, is the rest of the RNA in there a bunch of human RNA, in which case we would think, okay, probably wasn't from an animal. It was probably, from the handle of the cage, from the human who had COVID or SARS-CoV-2. But it also might be full of red fox DNA. And so we've been gently asking if they might share make those metagenomic data public, and we think we're making some progress, and we're hopeful that the Nature publication process will move that forward as well. So I'm hopeful that we've got lots more to find out. But yeah, I do want to emphasize that there's lots that we don't know still. Lots of important work to do. Hopeful that SAGO, this WHO group, will be able to collaborate with Chinese investigators and turn up more information. And I should say that our studies would not have been possible without the WHO investigation or study that was done. I think a lot of people criticize that effort, and I was actually guilty of this as well, as being far from perfect. And there's so much important data that actually is in there, including all of this mapping stuff that I was able to tap into, that was sitting there waiting to be drawn out. And so hats off to all the people who were involved in that, particularly the Chinese scientists who put that information together and are kind of unmentioned in a lot of these discussions.
I mean, I would expect the scientists would like to see the answer, but perhaps there is political pressure not to find out where it started, right? That would eventually confound any efforts.
I think there's very clear evidence that there is that kind of political pressure. I mean, I have been attacked personally after that Science Perspective for suggesting that the Huanan market was the epicenter of the pandemic by Chinese authorities. I don't think this doesn't fit their current talking points, but I hope that we can all actually come to a point where we let the evidence actually do the speaking. And I think certainly we're at the point where the Huanan market clearly was the epicenter. Some of the other details remain to be clarified.
So you made a point early in the first manuscript that this doesn't prove that it originated there, right? Or am I wrong?
I think we're at the point now where the evidence does show that the Huanan market was the epicenter of the pandemic, but we don't know many of the details, and formally, we don't know that it was that it got there through an infected animal or animals. But there's certainly a lot of evidence suggesting that that's how it happened. And again, this fundamental issue of all the 10,000 places it could rear its head in a city that size, it's at the place that you go to buy your live raccoon dog.
So early in the paper, you talk about the difference between an index case and a primary case.
Right.
And so we haven't told the listeners about the difference, and maybe you can do that. And then do you think that where we are, at least with an index case, that indeed that's what arose at the Huanan market? Or is that overstating it?
No, it's understating it. I think the primary case, so let me clarify what I mean, first of all. So index case, we mean someone we know about who's the earliest case in some cluster that we're interested in. in this case, we're just interested in, either the earliest case that we know about overall, or the earliest lineage A or the earliest lineage B case. The primary case is typically someone we don't know about who came earlier, but we didn't ascertain that person. We don't know, they slipped through, that maybe they were asymptomatic or just mildly ill. And so in our paper, we use information from the index case and the molecular clock and the epidemiology to estimate when the primary case, the actual first person infected, existed. And we do think that first person infected was infected at the Huanan market.
Okay.
When? Relative to the index case, first index case?
We think the estimates center on late November for that very first person infected. So quite recent, but again, it fits with the molecular clock that suggests only, December 10th for the common ancestor of the gene sequences. And with gene sequences, if the virus had been around since October or September in Wuhan, like some people think, you don't expect a time to the most recent common ancestor to be in December. You expect actually it to go back to pretty close to when the first case was. And so, yeah, I think we're hopefully people will start to understand that there really is no credible evidence for many cases at all existing before December, and any cases existing before November.
So when you mention these dates and the index case and the primary case, how does that tie into the lineage A versus lineage B story, just for clarification?
So there's actually a primary case for lineage B and a primary case for lineage A, because they're separate jumps. Just like HIV, we now know is multiple viruses that jumped from
M versus
animals. Yeah.
So in your manuscript, you make the statement that WHO mission members were told that no unlicensed or live trapped wild animals had been for sale at the market. And you provide evidence to the contrary with your photographs. And then you say later in the same paragraph, not a single raccoon dog from the market, live or dead, was tested, nor for those from wild populations. So why is this? Why do they say there are no live animals? Why didn't they test the obvious suspects? Is that an effort to obfuscate the origin, you think?
It's a good question. There's lots of possibilities. What does seem pretty clear is that by the time the China CDC got in there on January 1st, that there weren't any of the live animals that were photographed on December 3rd, or that were documented in November in the table in our study. Why was that? Don't know. But it's important that that's actually talked about, because there are people who try to oversell this idea that 80,000 animals were tested and we haven't found the virus. Well, the only ones that you really wanted to test weren't tested. And that's a pretty crucial point. Yeah.
So the fact that they closed the market, culled all the animals January 1, that says that they were worried about it clearly, right? So,
yeah, and again, they closed the market January 1st. Whether the animals were culled, I don't think I don't think there's evidence that they were culled as part of the closing of the market on January 1st. They seem to have disappeared before that. But anyways, absolutely, they were thinking SARS won, right?
Yeah, of course, sure.
And they're memorialized in print, in papers, in newspaper reports, basically saying, yeah, it's pretty clear that this is from illegal wildlife at the market. And that tune has changed since.
Do you think that by January 1, the virus was already out and it was too late to do that? Or did that have an impact on the subsequent outbreak, the closing?
I think whatever led to the removal of the live animals actually had an important effect in terms of reducing cross-species transmission events. Yeah.
So in the realm of speculation, I'm kind of going back to some earlier questions here. I'm trying to imagine whether both lineages entered the market as the lineages that they were, say in even one animal, or how much evolution might have gone on in the market itself. And I'm also interested in this idea that there could have been multiple different animal species involved, and wondering how that even could impact on the evolution of something that might have greater potential to spill over into humans.
Okay, so last part first. I don't think any of the differences between lineage A and lineage B were really important for the ability for these different lineages to spill over into humans. I think they both had that ability, and I think my guess is that the progenitors, the lineage A and lineage B progenitors, arrived separately in different animals at different times at the market, and that lineage B arrived first. That's a guess. There's evidence.
Arrived in people or in animals in the market?
Animals, I suspect,
okay.
but you can't actually rule out that people, say, working on one of these farms, were involved. You could even have a person who brought the infected animals from a farm and then stayed at the market being infected along with the animals that they brought that day. Yeah.
If A and B came to the market on separate animals, I would imagine that it would be most likely that those animals came from the same source. It's hard to imagine them coming from different sources. Yeah.
Oh, absolutely. Yeah. I think to me, the only scenario that makes sense is a single source, like a single farm transient outbreak, or a single local population, because of the shallowness. If they really are two jumps, they're from a shallow genetic reservoir.
So under those circumstances, you don't have to postulate a transmission chain in the market. There can be independent introductions from a single source into the market.
There can be, yeah.
Fascinating stuff. And I was thinking Jon Snow the whole time you were talking.
Oh, nice.
And I'm wondering, so you have timestamps on all these data points, right? I'd like to see this animated.
There's actually not great timestamps on these.
Okay.
For a few of them, yet, but actually that's one thing that would be great to eventually, that information exists, but not.
So that's why you have to kind of cluster it in a big group as before and after. That's sort of the resolution of your timestamp.
Yeah.
Okay.
Michael, let me ask you one more thing. Are all the markets now in China, all the live animal markets closed? Or if not, or if they are going to open at some point? The point is, so you can answer that. And then there are plenty of places in the world where there are live animal markets. So we're not going to close them all. What's the alternative? What can we do? So 2 questions. Sorry.
Yeah. I do think there was a pretty strong crackdown on live animal markets, at least selling illegal or illegally selling some species. There's certainly live animal markets that sell some animals live that is perfectly legal and regulated and is still ongoing. I think you'd be much harder pressed now to find a live raccoon dog for sale in places. And my understanding, I just learned quite recently from reporter Michael Standard that different cities had very different enforcement levels of regulations against illegal live mammal sales. So Beijing and Shanghai, for example, I think had much less open sales of live live mammals like we see in Wuhan, and again with Wuhan, with Hubei province where Wuhan is, having civets that were infected with SARS-1, very closely related viruses to SARS-1. There may be something to the combination of its loose regulation or enforcement, and so there's the opportunity for this to happen with the virus potentially circulating in bats locally in Hubei. So lots more to learn there. But okay, remind me of the 2 questions.
So assuming we can't close all the markets, there will be continue. What's the best other approach to preventing this from happening again?
I think we can actually clamp down on a lot of the most dangerous activity. I think, and I think China will be much more motivated to do that this time. Regardless of whether they ever, kind of face up to the evidence that this was very similar to what happened with SARS-1 and the methods and approaches that they had put in place to avoid a repeat of SARS-1 have failed. I think they're going to be really motivated to not have SARS-3 happen. And so I think you'll see a lot of crackdown in China, but China is just one place. There's lots of other places around the world. There are SARS-related viruses in Europe and Africa. And so one of the things that I think is important is that we're not going to be able to obviously shut down all of our ecological interactions with the animals that house this cornucopia of potential pandemic viruses. I think we can learn to limit them in some ways, but there's always a black market, even if you have the best laws and attempts at enforcement. But one lesson from what I've learned about the early days of the outbreak And, even going back to Zhang Jixian at the Xinhua Hospital, kind of figuring out on December 27th, the key things about this, that it was a new viral pathogen, that it could cause asymptomatic infection, and that it was human to human transmissible. So China had invested, I think, more than $1 billion in a pneumonia of unknown etiology program that was meant to catch things like this. But apparently it wasn't very well known to clinicians. And so it just wasn't being used very much. But if that system had been in place, and if there had been a more robust set of actions early on, I think it's, if the cordon sanitaire had been imposed on December 29th instead of January 23rd, it's possible that we wouldn't have had a pandemic. And so I do hope that one of the things that comes out of this is a global pneumonia of unknown etiology program, so that the ones that do slip through, we actually have a fighting chance of nipping them in the bud. I used to be a forest firefighter in British Columbia. I rappelled out of a helicopter, which is the more sensible British Columbian answer to smoke jumping, where you end up somewhere within a mile or two of the fire. But the whole idea of that program is you hit hard, you hit fast, you identify the fire and you get there immediately. And you put it out while it's the size of your bedroom, because you wait 10 hours and it can be the size of a city. And so we need to do that with these pathogens. We need to figure out how to do that on a global scale. And that's one thing that I'm not seeing discussed, and it's doable.
Michael, I have to tell you, I was reading this on the train yesterday, this manuscript, and I was so engrossed that I missed my stop.
I'll take that as a compliment.
All of a sudden they said the next town, and I said, what? I was totally engrossed, and it's never happened before. So this is fabulous. I hope it comes out soon. And
thanks. I'm putting that on my CV that you missed your stop.
And I want you back for the next one as well, okay? Because you said there's more coming.
Okay. Sounds good.
Thank you so much.
This was so cool.
Really appreciate it.
Learned so much.
Yeah. Michael Worobey, University of Arizona. Many thanks, Michael. Thank you.
Thanks, Michael.
Thank you.
Bye-bye. Oh, fascinating. Fascinating, right?
I would think that, the Chinese officials would certainly prefer this as an explanation to a lab leak. Okay, so might be interested. I don't know. Does
it would be better, wouldn't it? But I don't know.
But Michael's right. Over time, the truth floats.
Yeah.
So much for a series of questions, right? Boom.
Yeah. Right.
Yeah. So what the listeners don't know is that we had 40 questions in our doc. Some of them had multiple parts per question.
But I think we covered everything.
I think we covered it,
but in a really interesting way.
I mean,
my first question, all I wanted to know was how the authorship came together. But that
led to the whole thing.
Led to a whole summer. That's fine. Great.
Yeah.
No,
and also the whole time I was thinking about all of the stories that I've read from David Quammen about HIV's origins, and imagining the book that David Quammen or someone will write that sounds it's very similar based on this whole story.
He should. He's going to have to go visit the places, because that's what he usually does,
Yeah. Mm-hmm. I look forward to that book.
Boy, you have to wonder whether the fire jumping thing would actually work. I mean, the problem is the asymptomatic transmission. I remember, when we first started to talk about this, the first cases that arose were directly associated with the Huanan market. We said, oh, this is another one of these things.
Yeah.
And then a couple of cases came up that weren't obviously connected with the market. And so we said, oh, no, that's not the market. Okay. But there's the asymptomatic transmission.
Yeah.
It confounds the whole thing. And at the time, we didn't know about the asymptomatic transmission.
You would not expect them to be all at the market. I think he said that in his science thing. You wouldn't. Some of them are going to be outside. Right. Yeah, because of that. Yeah. Good stuff. I think I hope it gets accepted.
Oh, it will in some form. Yeah. It's got to. It's really great stuff.
Well, I got to tell you something. So the Mimi virus paper we did a couple of weeks ago?
Yeah.
Yeah.
So I sent it to the author, and she loved that. She listened. She said it was great. She said Nature rejected it after a year and a half.
Oh.
Because one of the authors didn't think it was exciting enough. They shouldn't have even allowed that kind of comment in a paper. Not exciting. I mean, I thought that was brilliant.
One of the reviewers said that. Is that what you said? I mean,
that's what she said. That one of them, because the other reviewer was from one of them. But a year and a half of your life is,
Yeah, I mean, that might be okay if there weren't a year and a half in between. I have actively discouraged people from going for big name journals. Sometimes because of that very problem.
It's crazy.
Okay? It's going to take a year, and then they'll tell you,
But I suspect in this case, they won't have any trouble publishing this.
No,
I sure hope not.
Because it's the thing. It's the story right now. So,
hey, it's already published. It's on TWiV, right? Actually, it's in the the media has been riffing on this some, right?
Yeah, but it will riff again once it comes out in Nature, right?
Yeah, but I think that hearing it from Michael Worobey,
it's great.
Sort of put this picture in my head of the mystery animals and thinking about the virus and the mystery animals. I just have such a neat model in my head now.
Yeah, even reading through the Worobey paper, which was more accessible for me than the 2Jump paper. Yeah. I became more and more convinced as I read through it. But then having him explain it, was just
good.
Everything.
For listeners, these preprints are available, and Figure 1 I found to be one of the most compelling, where he has this what's called kernel density estimate, which is these sort of shaded diagrams of where the foci of infection were. And that's where this before and after picture comes from. Okay. If you compare, for reference, Figure 1B and Figure 1E, that's the before and after picture. And it shows that the centers of those things are different. And it's quite interesting. It's very graphic.
Right. And also then they have a model in Figure 6, I think, of this Worobey et al. Paper that until he explained it, I really didn't understand or didn't have a good grasp on how within the Huanan market you get lineage A and lineage B going to people from some, So Rich's questions really helped clarify that. So
I think there's a little bit of sleuthing involved here, right?
Oh, man.
Right? Mm-hmm. For trying to find the density of the home addresses and also the pictures of the animals that somebody took in December.
Well, and he said in his, he said as he was introducing the whole thing that, I mean, one of the things I got out of his introduction is that there's been a lot of essentially anecdote, little points of, well, it could have come from here, it could have come from there, it could have come from this cave, could have come from that cave. And he said, dude, we need real data, okay? Take all this stuff and go over it with a fine-tooth comb and do the appropriate epidemiology so that now we have data rather than a bunch of anecdotes.
Yeah, I'm convinced.
Yeah, I'm totally convinced.
For sure.
Okay, let's do some picks. Brianne, what do you have for us today?
So I have a place that I visited last week at this time, and I thought was appropriate because I was in Tucson, where our guest, Michael Worobey, is from. And it is actually a set of caverns that houses some bats called Karchner Caverns that I was able to visit. It's just an absolutely fabulous place that I recommend anyone go to visit. It was the second time that I'd been there. It's a living cave. So the structures are actually still sort of in the process. You have to worry a lot and think a lot about whether or not you are bringing in microbes to influence the cave. I got to see lots of areas where bats had been roosting. They're not currently there. And it was also just beautiful to think about how caves like that form, look at the fossil deposits and see how they form and what they actually would look like in a cave. And also to think about how these two, I guess, then graduate students sort of found a sinkhole and decided to go in it. And found this amazing cave system. And so all of the parts of the story, I think, are cool things for scientists to think about. And I had a great time. And I recommend it to anyone.
This is near Tucson?
It's a little bit south of Tucson, yes.
I think I went years ago. This is the one where you have to make an appointment. They only let so many people in at a time. Is that right?
You do have to make appointments. There are two different sections. I did one section the last time I was there a couple of years ago, and I did the other section this time, and it's fascinating.
Yeah, they don't want too much human breath in there and that sort of thing. Right? Yeah,
yep.
I remember
and
that.
And if you happen to touch anything while you're inside, you need to tell the person so that they can flag it, so that they can come back and wash it with cave water to get rid of any oils or microbes that you've deposited where you've touched.
Yeah, it's cool. It's very cool.
So I've just bookmarked this under my travel folder.
Yeah, you got to go there. It's cool.
If you go there, Rich, I'll tell you a whole bunch of cool things in the area.
Okay, good. This looks like a extended family expedition. Guys, see, they got cabins and stuff.
Yep.
Cool.
Kathy, what do you have for us?
I picked two YouTube videos. I think we've picked both of these guys before. One is the Smarter Every Day series, and the other is Veritasium. And I don't know how I happened on the first one, which is, the full name is Why Laminar Flow is Awesome. And it's just fascinating. He's got a swimming pool, like a kid type swimming pool, and he pokes holes in it, and you look at laminar flow, and he just goes on and on about it. And then it's answered by Veritasium. Turbulent flow is more awesome than laminar flow. So the two of them together make a really fun dive into laminar flow and turbulent flow. And turbulent flow has a Chris Hadfield cameo at the beginning of it. But I just highly recommend those two.
Excellent. I'll have a look at these.
And clearly, they're in conversation with one another because, the titles match and that kind of thing. So, yeah, check it out.
Rich, what do you have for us?
This may be a little mundane, but I can't help myself. It may also be a repick, okay? But I want to plug Wikipedia by picking Wikipedia as my pick, okay? And I didn't pick. I decided not to pick just the Wikipedia homepage, but the Wikipedia page that describes Wikipedia.
Yeah,
okay?
yeah.
So you can read all about its evolution and all about it. But this, I must access Wikipedia at least a half a dozen times a day, and probably more. And as far as I can tell, it's an extremely reliable resource, and it's the world at your fingertips. It's unbelievable. Okay. And it doesn't have any advertisements. There's nothing. Okay. This is entirely, it's mostly volunteer. There are a few paid individuals, but mostly it's a bunch of nerds typing stuff up on a volunteer basis. It seems to be quite well supervised. Of course, I wouldn't necessarily know when I find inaccuracies, but when I look up things I know about, they're accurate to a very large extent. And so in the plug realm, when occasionally I go there and they are asking for money, I give them money because, I get more out of this than a lot of other things that I get money for. And I think it's just a wonderful enterprise. So there you go.
Yeah, I always donate too, just to get rid of the thing at the top of each page. It's a good cause. My pick is a article by Carl Zimmer called "The Human Lake." It's at Discover magazine in his column, The Loom. It was published in 2011, but I just heard of it from Mark Martin, who was on TWiV last week, and we had a letter from a professor at a small college who she said she had to start teaching ecology, and she didn't know anything about it. So where should she start? And so he said, one of the things you should do is read this article by Carl Zimmer. So I went and read it, and it's great. It's about Lindsay Pond, which is a lake in southern Connecticut where G. Evelyn Hutchinson used to work. If you were to have gone to the lake 70 years ago, you might have seen a gentleman swimming across the lake holding a container of radioactive phosphorus, which he then dumped into the water. So apparently Hutchinson was the founder of modern ecology, right? A science based on theory, math, asks fundamental questions rather than just cataloging stuff that's out there. So this is an article all about that. And so I like it. It's really good. It's a great introduction to ecology.
This looks great.
That looks fantastic. I want to send it to all my colleagues.
And we also have a listener pick from Brad. I've been watching your YouTube channel for about a year. Would like to say thanks for your quality information, which even a person without a college education can understand some of. I found TWiV after seeing you on Lex Friedman, whose shows I enjoy a little less now. I hope this is a simple question. Is there a specific episode that points out the inaccuracies in the information Peter McCullough and Robert Malone are putting out, or is it over a number of episodes? If so, which ones? So we haven't addressed that, but apparently there was another podcast where they did, and we talked about it. Someone picked it on a recent TWiV. So I will see if I can pull the link for you when I put the show notes up, Brad, because they apparently did a point-by-point analysis, and there's no need for us to do that again. The document More Harm Than Good by the Canadian COVID Care Alliance has been making the rounds in my place of work, and I am not knowledgeable enough to answer the falsehoods in this document. Thanks to you and everyone else on the panels for the great information you share and for your picks of the week. I have enjoyed the ones I have visited. My pick would be another YouTube channel, NASA Spaceflight. Channel is a lot like Microbe TV in that they are supported only by donations, and they use their own cameras to cover rocket launches around the U.S. live on YouTube. I first found them when SpaceX was doing a lot of Starship test launches in Texas. There you go, Rich.
Cool.
Check it out.
Yeah, I will.
That is TWiV 876. You can find the show notes at microbe.tv/TWiV. You can send your questions, comments, picks to TWiV at microbe.tv. And if you like what we do, we don't put ads on YouTube. We depend on your support. We don't do any more ads. We would love to have your support. Microbe.tv/contribute. Rich Condit is an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
And Brianne Barker's at Drew University, BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here. I learned a lot.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back soon. Another TWiV is viral.